Hi, my name is Susan Howell, and I'm the genetic counselor for the Extraordinary Kids Clinic at Children's Hospital Colorado, as well as a board member of AXYS. And I'm here to speak with you today about genetic counseling for X and Y variations, and specifically understanding XXXY. So to begin, in regard to some of the shared terminology, just so we're all on the same page, when we use the word chromosome, we're talking about the structure inside of a cell that carries our DNA or our genes. And these genes tell our bodies how to grow and how to develop. When we use the term aneuploidy, we're talking about additional or missing chromosomes that can be present in the cells of the body. When we use the phrase sex chromosome aneuploidies, or SCA as an abbreviation, we're talking about specific X and Y chromosomes that are additional or missing in the cells. The term sex chromosome trisomies refers to conditions in which there are 3 sex chromosomes. So we can see Klinefelter syndrome with XXY or an additional X chromosome, Jacob syndrome or XYY with an additional Y chromosome, or trisomy X in girls with an additional X chromosome. And then the term sex chromosome tetrasomies refers to the presence of 4 sex chromosomes. And today we'll be talking about 48 XXXY. So collectively, these X and Y variations are given many different names, but for the, all intents and purposes, they're all relatively the same. Sex chromosome aneuploidy is a term that's more commonly used in the scientific community and in the medical literature, but we can use the terms sex chromosome abnormality or anomaly or disorder or variation. And then there's also the term of X and Y chromosome aneuploidy. And X and Y chromosome is really more straightforward in understanding, more logically. And then X chromosome variation is a term that's commonly used by families and often preferred by families that we work with. But when I'm talking about XXXY, a lot of times these X's and Y's get jumbled, right? And so some people will call it XXXY or XXXY syndrome. Some people call it 48 XXXY because that represents the 2 additional chromosomes. I tend to use the term kind of 3XY or 3XY syndrome because I feel like that's a little easier to roll off the tongue. And collectively, if you imagine that in this picture that the size of the symbols really represents how common these conditions are, we see the trisomies in the center that are much more common than the tetrasomies, but there are a lot of different variations that we can see in the population of the X and the Y chromosomes. And collectively, these conditions all occur in about 1 in 400 live births. So they're really common in our population, with the trisomies being the most common. When we're talking about 48 XXXY or the 3XY condition, it's about 1 in 18,000 males who are born with this condition. And to give a little bit of history and background information, is this diagnosis is often brand new to a lot of families. They've never heard of this. In 1956, Dr. Jacobs actually identified chromosomes for the first time under the microscope. So they're not conditions that have been around for too long. And we learned a lot from the newborn screening studies in the 1970s about the conditions with newborn screening, although the prevalence being most of these conditions that were followed over decades were trisomy conditions. When we look at the amount of information that's available in the medical literature, there really is a dearth. When we look at 48 XXXY as far as a syndrome and how often we're seeing it in publications in total, so going way back in time, there's only 80 publications about this condition in the PubMed Lit search, compared to like trisomy conditions that may have 600 or 900 plus publications. So there is so much work that needs to be done and publications in order to increase awareness and knowledge about these conditions. Even in years where we had the highest number of publications going back to 2020 with COVID onset, those were only 5 publications in the highest year. So we really have a lot of work to do around publishing in this condition. So how is this condition identified? Historically, for the most part, these conditions were identified in individuals that presented with congenital abnormalities or birth defects. They can be slightly more common in this patient population. They also are often patients are identified when they present with significant global developmental delays or low muscle tone. This diagnosed as hypotonia. And then autism is a common indication for genetic testing. And so often there's ascertainment through workup after a diagnosis of autism. But these conditions, 48-3XY is much more common to be identified because of the fact that compared to, for example, XXY, they're definitely more significant of a clinical presentation that we'll see in our patients as far as all of these areas of development and medical and physical features. And so that does promote earlier ascertainment. But a lot of the features that we can still see with this diagnosis, such as ADHD or anxiety, don't trigger genetic testing. There's not an indication for genetic testing when you have a diagnosis of ADHD and or anxiety. In 2013, there was a new test that was introduced commercially called noninvasive prenatal screening. And this is really identifying a lot of these conditions today. So non-invasive prenatal screening, also known as NIPS, is a blood test of the mother's blood in which there are fragments of DNA that are derived from the fetus, actually specifically from the placenta of the pregnancy, and then they're naturally found circulating in the maternal blood. And so we're able to screen for these conditions, including 3XY, by using this blood test during pregnancy. So it's also a test called Cellfree DNA testing. And as of 2020, the American College of Obstetrics and Gynecology recommended that all women, regardless of age or background risks, be offered this type of test because there was no risk in drawing mother's blood for miscarriage. It is dependent on gestational age, that the pregnancy needs to be at least 10 weeks, and that's because we want the concentration of those fetal fragments, or the fetal fraction is what it's called, to be high enough to be able to be a good screening test. Possible results of this test include a positive result, a negative result, or can be inconclusive. And there's a lot of different approaches around how this technology is utilized based on the lab. It's not a diagnostic test. So this test is a screening test that shows an increased risk potentially of a condition. But what we've seen in the patients that we've followed with 3XY is it's really not a result that's validated by most of these labs. So the 3XY diagnosis is usually identified with discordance from the result of NIPS being positive for an increased risk for XXY or XYY, but when in fact there are actually 2 additional chromosomes on diagnostic testing. So diagnostic testing is really important when we see an increased risk based on NIPS. So how is diagnostic testing performed? Prenatally, it can be done either by a sample of the placenta or amniocentesis. Amniocentesis is a more preferred method of doing prenatal diagnosis with an NIPT result because of the fact that it's not testing the cells of the placenta like CVS does, but actually testing the fluid from around the developing baby. And it's usually done at 15 weeks or later in gestation. And then the postnatal test that we can do is either a blood test or a cheek swab or saliva test of an individual. And so either one of these samples are analyzed by a technique called a karyotype or a microarray, which is studying the chromosomes, and it's diagnostic to confirm whether or not an individual has 3XY. So what is a karyotype? So if we open up a cell in the body, we would expect to stain the chromosomes and see a picture kind of like this at the top of the page. And then the lab puts the picture in an organized picture depicting the pairs of chromosomes in an individual. So we typically have 23 pairs of chromosomes, and they're numbered 1 through 22 from largest to smallest. And then our sex chromosomes are labeled with either an X or a Y. And we have pairs of chromosomes because we get 1 chromosome in each pair from each parent. So we get 1 from mom and 1 from dad, 1 from mom and 1 from dad. So typically there's 23 chromosomes in the egg and 23 chromosomes in the sperm. And at the time of conception, then we have 23 pairs or 46 total chromosomes. The Y chromosome carries the genes that drive male development. And then in the absence of the Y chromosome, when we have 2 X's, then the development is as a female fetus. When we diagnose 3XY, we see a picture that looks like this. So this, you can see the 3 copies of the X chromosome and then the Y chromosome. And so this is a diagnostic test by Karyotype confirming a diagnosis of 3XY. The other test that can be done is called a microarray. And so this test looks at each individual chromosome pair, so the number 1, the number 2, and 3, and so forth. And it's using probes that attach along the length of the chromosome and demonstrate copy number. So in this depiction, a 0 represents 2 copies of the first chromosome and 2 copies of the second chromosome, all along the length. And what you see at the end here is an overrepresentation of the X chromosome. And so this would represent, if this traditional line is 2 copies of each chromosome, then this would be an extra copy or a third copy of the X chromosome that you would see in a microarray result that would diagnose 3 X's present in the cell. So what causes 48XXXY? This condition is a result of the chromosomes not dividing properly during cell division. And most commonly, it's a result from the formation of the egg or the formation of the sperm, in which there's extra chromosomes present at the time of fertilization from the egg or the sperm. We can also see mosaicism that results from a kind of a post-conception non-disjunction. So the egg and the sperm have what they're supposed to, and that the chromosomes fail to separate properly during cell division after fertilization. But it's not caused by anything that a parent would have done or not done either before or during a pregnancy. And this is not something that parents can blame themselves for because it's not your fault. We do see a lot of variations within this condition. So there's variable physical features that we can see, developmental features that we can see, medical features. The most universal feature of 3XY is testicular problems and the infertility, as well as testosterone deficiency. And then we can also see variable psychological features impacting learning as well as behavior. So this is a relatively busy slide, and I could make a talk just on this topic alone. But I did want to take a table from one of our publications in talking about 48-3XY to really explain kind of how it is similar, but with overlapping features of XXY, but it really is not a variation of XXY. And a lot of that has to do with the fact that it's much more rare than XXY. It's approximately 1 in 17, or I said 18,000 earlier, depending on the study you read. The number or the percentage of expected cases that are actually brought to clinical attention during an individual's lifetime has never really been studied or published. And there's not any data about kind of the age at which individuals are most commonly diagnosed. What we do know is that tall stature tends to be a feature of this condition, with the average adult height in men to be about 6'3". Obviously, height is heavily influenced by our parents' height, and so our background genes, but we do see taller stature in this diagnosis. We can see a variety of physical features The first one being clinodactyly, or curvature in the pinky finger that's represented in the picture here. We can also see radio-ulnar synostosis. So this is fusion of the radius and the ulna in the forearm. And we see this most commonly impacting the rotation of the arm. And so this is part of our clinical assessment, is to see if someone can fully rotate their hands from palm down to fully palm up, because this fusion in the bones can impact that. We can also see the presentation of some bowing in the forearms that represent the underlying fusion. We can see more common hernias in the inguinal area, as well as some other birth defects and congenital malformations that have been previously published and associated with other conditions as well. We do universally see the testicular problems associated with infertility and the inability to produce enough testosterone. Those are kind of individual testicular problems. Testosterone treatment does not cure infertility, so it's a different problem with the testes, but we do universally see that as a problem in our patients. We can see a variety of developmental and cognitive impairments in our patients. More than half of patients with 3XY will need speech and motor delay therapies. And we do see about 100% with learning disabilities, of which more than half will have a full-scale IQ, typically around 40 to 75 points. So to kind of contextualize that, I've included this picture where it shows you the bell curve of the general population and an IQ, and the average IQ is 100, and the colors represent standard deviations out. So 70 is considered the line in which a diagnosis of intellectual disability is confirmed, an IQ less than 70, and then 70 to 80 tends to be kind of this borderline, or 70 to 75 tends to be more of a borderline. So the majority of individuals with 3XY have an IQ of 75 or below, and we usually see areas in which their verbal IQ is more impacted from the speech and language and reading abilities than their performance IQ or their spatial and math. We often have patients that have a lot of really good spatial skills and can tell their parents where to drive from a very young age just based on memory, on being in the car and going to different places. And then as far as kind of origin, we see that 3XY results half the time from the extra chromosomes being present from the egg, and then the other half the time being from the sperm. But it's important to keep in perspective about these conditions. So there's obviously a lot of variation, which I've just covered, and we still have a lot to learn from the children who are being identified with 3XY as a result of this new technology in NIPS. So we're doing that as part of the Extraordinary Baby Study, because some of these children were initially identified as having a trisomy and then confirmed to actually have a tetrasomy. We also recognize that family background genes and the environment in which children are raised have a significant impact on their outcomes, and can really attribute a lot of the successes and strengths that we see in our patients. We know that taking a proactive approach, especially when a condition is identified early, or prenatally especially, can be very beneficial. And then just keeping in mind that children are individuals, right? And as individuals, we all have our strengths and we all have our weaknesses, and we all have those areas that we need to work harder on and that come easy to us. And so, recognizing that this diagnosis does not define a child is really important. And that we see a lot of these strengths and beautiful characteristics in many of the patients that we see and follow in clinic. So in working with genetic counselors, I just want to make sure that people are aware that genetic counselors can be helpful in a lot of different areas, not only explaining a diagnosis, but then planning next steps with a family after a diagnosis is made. Sometimes it's attributing various symptoms to the diagnosis, or even recognizing that they're not attributed to the underlying genetic diagnosis and warrant further evaluation. Genetic counselors can be helpful from disclosure of the diagnosis, how to talk to the child about their diagnosis, how to talk to family members or the school about the diagnosis. Also helping kind of advocate in the school district, recognizing that learning disabilities are very common in this patient population, and being able to teach a new family about how to navigate the school and advocate with the school, as well as navigating healthcare if there are health concerns or presenting symptoms that I've attributed to 3XY. And then also talking about transition to adulthood and what that might look like for an individual and how to set it up for success. Genetic counselors can be very helpful in connecting with community resources, as well as addressing any kind of recurrence risks based on affected individuals. There's a lot of wonderful resources available through AXYS on the website. And while there may not necessarily even be specific to 3XY, I do think that the resources that are available, even if they're referencing XXY, can still be very informative to families. Understanding testicular functioning, how the treatment works, can be very similar in 3XY. The Extraordinary Kids Clinic that we run at the hospital has a wonderful website as well, with a lot of publications that are available for download for free. And then Amazon has many publications. We created a series of books. Unfortunately, it does not address specifically 3XY, but we are working on additional resources. So keep your eye out for that on Amazon as well. And with that, I'm happy to take any additional questions you may have.